a brand new episode that I like to call Making Something Out of Nothing. We're going to talk about DNA versus RNA, translation versus transcription. What you got with the messages and with the transfers. All right, people, so let's get started. When we start this entire thing off, we got to think to ourselves that we are all made of this stuff called DNA. And the idea then is that the DNA is coiled up and put into a chromosome. Now, along these chromosomes, there will be little pieces, little segments, right, of each part of this chromosome that will actually code for a particular trait that we will be able to physically see or will have inside. And the idea really is that those things that code for the traits are called genes. Now, genes need to be expressed. This is how proteins are made. And so to go through this particular process, we have to go through two steps. One thing called transcription and the other one called translation. Instead of opening this thing up and then literally splitting it all the way into pieces, the way that we did when we were looking at DNA replication. We had helicase and DNA polymerase, and that split apart that particular DNA strand. Now, instead of doing that, we're not gonna just pull the whole thing apart. We're just gonna go for a tiny little piece and get that particular gene. So the DNA will be able to open up, and then we'll focus on one little strand and we'll rip that thing open, and then we'll be able to try and take a code and the genetic instructions to make that particular trait. And the way that we do it is we do it through some helpers. Now, RNA polymerase is going to be our biggest dude. And RNA polymerase is going to be the particular enzyme, ACE, A-S-E, meaning enzyme, polymer, long chain of macromolecules, and RNA, right? RNA polymer ACE. It's an enzyme that makes a polymer of RNA. And it's going to, as it unravels that little piece of DNA right there and opens it up, it's going to start to read a single line of that DNA, a single strand. And it's going to create another copy of instructions from that strand, where it's the gene that we want to express. Now, the deal is that we are looking at this new strand being very, very, very different from DNA. Why, you say? Well, because the idea is that DNA is double-stranded, but RNA, the stuff that we're starting to make, right, is single-stranded. Not only that, but A's used to equal T's and C's used to equal G's for DNA. And the idea now is that with RNA, A's equal U's and C's equal G's. So the big difference right there is that our thymine, the T that used to go with A, is now going to be replaced with U, uracil. The idea of why this is so important is now the cell can recognize what is a copy and what isn't. Remember, if it's copied, there's a U and there's a single strand on it, then the nucleus says, cool, you're allowed to leave, right? It's like showing up to the costume party with the costume. You get to go in, right? But the deal is if you are DNA and you're double-stranded with T's and A's and C's and G's, man, you better not be going out of that party, because the other thing would be if we change that DNA at all, then now all the cells that we're going to make after that came from pre-existing cells. So now they've all got this crazy change going on, and that can't happen. So we're at the point where RNA has been made from a DNA strand inside the nucleus. This single-stranded RNA molecule, ribonucleic acid, is going to be able to exit the nucleus, and it is carrying a message. So we like to call this, folks, mRNA. And now, if we take that mRNA and we move it out into the squishy, gushy, ushiest good stuff of the cell called the cytoplasm, that is where we're going to start seeing this particular code get read. Now, to read the code, we need a special helper. And that special helper is going to be our transfer mRNA, the tRNA. This is going to create a process called translation. It's going to help it go down. Now, what's doing the translating? Well, really, the thing that's going to be making the particular amino acids, which are the monomer of our proteins, the thing that's going to be making those particular amino acid polypeptide chains are going to be our ribosomes. So this ribosome is going to be able to fit onto that particular little strand of messenger RNA that just moved out of the nucleus. And then we're going to have these awesome little codons that it's going to be reading. It's going to be reading these single-stranded little letters that we know as nucleotides, right? These nucleotides are going to get read, and a complementary base pairing of these nucleotides is going to be attached to it. We call this complementary base pair of the codon, the code that was written down during transcription. We call that complementary base pair that is starting to help us figure out how to translate stuff, the anticodon. And the anticodon is what tRNA is going to help out with. It is going to lock these things like little locks and keys, or like little puzzle pieces. They're going to be able to latch on to one another, and then they're going to be able to communicate what the amino acid is and in what sequence the amino acid is in, so then we can properly make that protein.